Now, most biological systems are regulated through some sort of feedback mechanism. And these three feedback mechanisms basically allow self-regulation. And there's two different types. You can have negative feedback, and negative feedback means that as you go from whatever you start with to whatever you're ending with, and these levels build up more and more, it basically shuts down the process so that this is not being converted to that. So as something builds up, negative feedback shuts it down. Now, it doesn't have to just be chemical reactions in creating products. This can happen through, different processes in the body. So for instance, if you had your body temperature increasing, so here's your set point, let's say it's at 98.6 degrees Fahrenheit, and let's say your body temperature was up to 101, well, one of the things that would happen is that you would start sweating, and the reason you sweat is to bring it back down. So since you're trying to go against that rise, that's called a negative feedback. So if you stop what's changing, it's a negative feedback. On the other hand, positive feedback is going to encourage what's changing. So when you have product being converted or reactant being converted into product, as that product builds up, it will actually enhance and make it happen faster and faster and faster. So you get more and more product. If we looked at a process in our body, if you think of something like childbirth, when a woman goes into labor, as the baby starts moving in the uterus, the wall of the uterus stretches and that causes muscles to contract. That also releases oxytocin. Oxytocin stimulates contractions to be harder. That releases more oxytocin, which stimulates more contractions. So when a woman goes into labor, what you'll notice is the contractions get closer together and they get stronger. And it's basically amplifying the signal by releasing more and more and more oxytocin. So if you have more and more and more, it's a positive feedback. If you're trying to stop what's changing, it's a negative feedback. So in this generic example, we have some sort of A that gets converted through B, C to D. And as D builds up, it basically stops this enzyme from working. If you stop this enzyme from working, you create a roadblock here, which means A can't be converted to B. No B means no C. No C means no D. So if you build up D and there's plenty of this and then it stops this, that's a negative feedback. In this case, you have W to X, X to Y, Y to Z. Z starts building up, but Z attaches to this enzyme, and we haven't talked about enzymes yet, but enzymes help control the reactions in our body, and that's going to enhance this and make it happen even faster, which means more Z gets built up, so that's a positive feedback. And one of the things I started off with in this chapter is the idea of evolution, and evolution is how things change over time. And evolution is one of those core themes in biological understanding. So Theodosis Dobzhansky said nothing in biology makes sense except in the light of evolution. And evolution unifies these different scales of biological processes and living entities on our Earth over the history of Earth. And billions of years of evolution have produced diversity of different organisms. So we're going to start with some basics here. But once again, if you go into Bio 102, we'll do an entire chapter looking at the history of life on Earth and talking about evolutionary processes. So when we look at biodiversity on our planet, which is the number of different species over given areas, there's approximately 1.8 million species that have been identified and named to date by scientists. The thing about that is that thousands of species are discovered every single year. So there's been years where the number of species range between 18 to 35,000 species, which is amazing to think about because I think most people tend to believe that we kind of know every nook and cranny of our planet, but that's just not true. And I'll put an article up on the Blackboard that I want you guys to look at, but it just highlights the fact that we're discovering new species all the time. And yeah, sometimes it's bacteria and little things that would be hard to identify, but oftentimes it's even reptiles, amphibians, and mammals. So the question becomes, if we're discovering new species every year, how many species are on our planet? And there's ranges of estimates that are between 10 to 100 million species. Now, most mathematical based models are kind of agreeing down around this lower end. But with that being said, let's round it up and say we know 2 million species on our planet. That means there's still, 8 to 10 million more species on our planet that are left to be discovered. And that's amazing to think about because there's a lot of unknowns out there. And when you look at this idea, there's kind of a downside to it also. You guys realize that there's also species going extinct all the time, which means there's species going extinct that we don't even know are here in the first place. And that can create a large level of uncertainty in how our ecosystems function, and even for the future, the potential to benefit us in the sense of medicines and things. So once again, we'll come back to some of this in Bio 102, but it's an important concept to understand that we're discovering new species all the time. But since there are so many different species, we have to have a way to kind of categorize them and organize them. And that's where taxonomy comes in. So this is a branch of biology that basically groups organisms into increasing breadth of categories. So we're going to have domains, which are followed by kingdoms, and they're the two kind of broadest units of classification. And we're going to break this down even further because you're going to have to know the levels of classification. So domains, kingdoms, phylums, class, order, family, genus, species. And I'll show you that, but that's how you're going to have to understand how to organize things. So if we take an example, first of all, you have a domain. So the domain in this case is eukarya, and eukarya is everything that's made out of eukaryotic cells. And that includes protists, fungus, plants, and all kinds of animals. But that domain is a big category, which means if we want to kind of narrow our options down, then let's take domain and break it up. That's going to be kingdom. So in this case, we're looking at the Animalia, which means just animals. Now, you don't have to know these terms here. This is just an example. You will have to know these terms up here. That's what you're trying to pick up on and learn here. So when you take the kingdom, that kingdom can then further be broken down into the phylums. So in this case, the phylum is Chordata. So the chordates are animals that have a notochord and pharyngeal gill slits and all sorts of things we'll talk about in the future. Phylums are broken up into classes, and the class here is Mammalia, which includes the mammals. And then the order here is Carnivora, which are the carnivores. Family, which is Eurycidae, which is referring to bears. The genus Eurysus, which are true bears, and then finally to a species, and that's one single organism called Ursus americanus, or the American black bear. So this, once again, are just examples. You don't have to know them, but you do have to know this. Domain, kingdom, phylum, class, order, family, genus, species. You have to know that level of organization. So when it comes to the domains, there are 3 domains you guys have to know. You have the domain bacteria, the domain archaea, and these are all made out of prokaryotic cells, so these are prokaryotic organisms. And then you have the name eukarya, or the domain eukarya, which are made out of eukaryotic cells. These are eukaryotic organisms. So there's the domain bacteria, which are bacteria that you might commonly understand and know. There's the domain archaea, which many people are not familiar with. They're bacteria-like, but they are different, which is obviously why they're in a different domain. And then you have the domain eukarya, which includes things like animals, plants, funguses, algae, protists. So you have to know those 3 domain names: bacteria, archaea, and eukarya. Now the domain eukarya is broken down into 3 kingdoms. You have the kingdom Plantae, that includes plants, fungi, which includes the mushrooms, funguses, and then Animalia, which are the animals. So they're the 3 kingdoms you know. So some of you might be familiar with another one called Protista, but Protista isn't really recognized as a kingdom. They're kind of categorized based on how they fit with plants and animals. But this is a 3 kingdom system, so you have the 3 domains. And when you look at one of the domains called eukarya, you have to know the 3 kingdoms that make that up: Plantae, fungi, and Animalia. Now, even though we have all that amazing diversity on our planet, between all those living things, there's remarkable unity there. So when you look at that underlying unity in the diversity of life, you see things like DNA is the universal genetic language that all organisms have, from bacteria all the way up through animals like humans. So if you take bacteria if you take DNA from a bacteria, or DNA from a plant, or DNA for a human, and you insert that into another organism, they can read that DNA because it's the universal language of living things. And then you also see unity in the evidence for features of cells. When you look at different cellular structures, that's a common hold throughout the evolutionary process. As an example, this is a type of cellular extension called cilia. Cilia are these hair-like things that stick out of the surface of some cells, and they're typically used for sweeping or moving or allowing the cell to swim. So here's a paramecium, and you see the cilia all over the outside of it, and that's how it swims through the water. But here's a cell, one of the cells in your windpipe called a pseudostratified ciliated columnar epithelial cell, and they also have cilia on it. And when you look at the cilia from human cells or the cilia from paramecium, they have this exact same protein arrangement inside of them. Now, what you have to realize is the history of our planet, we have amazing diversity that's changed over time. And obviously, some of that's been lost, like the dinosaurs, for instance. The way we know that is by looking at fossils. So the fossils provide us a glimpse into the past because the fossil record, first of all, isn't complete, and not everything that's lived on our planet has fossilized. But the fossils do show us evidence of how life has evolved or changed over billions of years. So Charles Darwin is known as the forefather of evolution, and he is not the only scientist that worked on the evolutionary idea, and he's not the only one that came up with the idea of evolution, but he was one of the first ones to write about it. So Charles Darwin published On the Origin of Species by Means of Natural Selection in 1859. And in this, he made two main points. Number one that you have to know, species show evidence of descent with modification from a common ancestor. In other words, as you go from generation to generation to generation, there's slight changes that occur. And also, natural selection is the mechanism behind descent with modification. In other words, the reason things change over time is the best fit with their survival in the environment. So Darwin's theory explained the duality of unity and diversity. In other words, how can we have so many different species on their planet, but there's still certain features that they share? So there's the duality there, meaning they're both together. There is unity, but there's also diversity. So if we look at this picture, for instance, these three flowers you're looking at are orchids. Now you look at them and there's amazing diversity there. They're different colors, different shapes, different sizes. But you also notice that there's some similarities between them. They have, these petals that come off the bottom. They have these flanking side petals. They have this top cap petal. So the idea there is, yes, there's diversity. They look different, but there's also unity. Their overall base structure is very similar to each other. And that's what I mean by that concept that there's duality in diversity and unity. So in order to come up with those 2 points, Darwin made observations. One of the observations he made is that individuals in the population have traits that vary. Now, if you think about that, that's pretty obvious. If we were sitting in a classroom and you just look around, we're all individuals of the same species in that room, so we're a population. However, we all have our different characteristics. Some of us have different hair color and eye color and different heights, left-handed versus right-handed, different tones of our skin color. So there's just different features about us that make us individuals, and we have varying traits. And if you think about point number 2, where did we get those traits from? We got those traits from our parents. So your parents passed those traits off to you. Now, that doesn't mean that you're identical copies of your parents because you are genetically unique, and we'll talk about why that's possible when we do genetics later this semester. But even though individually we all have varying traits, we got those traits passed down from our parents. More offspring more offspring are produced than survived. And what that means is that when offspring are born, not all of them are going to survive. Now, humans, people often think about that's just something that happens. Our offspring survives. And it's because of modern medicine. But in actuality, that's obviously not true. But just to understand this better, we can look at other populations. If you had an insect that laid, 150 eggs, how many of them are actually going to survive? If you have a sea turtle that came up to the beach, laid 100 eggs, how many of them are going to survive? Not all the offspring survive. So more are produced than can actually survive. Another observation Darwin made is that competition is inevitable. Things have to compete with each other for survivability. They have to compete for space, for air, for nutrients, for food, for sunlight, for whatever it is. So competition is something that all living things face. And species generally suit their environment. In other words, they're adapted to their environment, which is why if you go around the world to different environments, you find different species there. There are some species that can survive basically everywhere, but that's pretty rare. Usually organisms are ideally adapted for their environment. Now we could take that to an extreme. Like if you look at the plants in the desert versus the plants in the tropical rainforest, they're very different. And if we took plants from a tropical rainforest and put them in the desert, they most likely wouldn't survive, and vice versa. Desert plants most likely wouldn't survive the tropical rainforest. But that's because they're suited, they've evolved to fit those environmental requirements. Now, Darwin also made inferences. So he inferred that individuals are best suited for their environment are the ones more likely to survive and reproduce. So if you're able to survive in your environment, you surviving means you have a chance of reproducing and passing your genes off. Those individuals that are not adapted to their environment will likely die. If you die, you can't reproduce, which means you don't pass those genes on. So over time, more individuals in a population will have the traits that enable them to survive. And that's an inference Darwin made based on the observations. And that's the idea of natural selection or survival of the fittest. It's the environment that selects for the beneficial traits. And realize when you say environment, it's not just the air and stuff around you. The environment is everything. It's the predators and preys and food sources and temperature and everything. That's selecting for who survives and who doesn't. So here we have a population of beetles, and in that population, there's variation in beetle color. There's every, dark to light coloration. But now we're going to introduce a predator into that population. So based on that environment, which is very dark, and you have all these beetles there, now comes the selective pressure of a predator, and the predator starts eating beetles. But you'll notice that it's easier for this predator to find these beetles and eat them because these ones blend in more with their background, so it's harder to see and capture them. So over time, if those predators are selecting against the lighter color beetles, that means that the darker colored beetles survive, and over generations, more and more of the population will shift to darker coloration because of that being a successful trait in that environment. So Darwin's idea of unity and diversity is going to rise from descent with modification. When you have a common ancestor, and over time, the environmental pressure causes those common ancestors to shift around, leads to new species, obviously there's going to be some of the characteristics that they all hold on to. So for instance, and these are just examples once again, but if you look at the forelimb, which is your forearm of a human, a bat, or a horse, or a whale, they all have very common skeletal architecture. So for instance, in your forearm, there's two long bones, the radius and the ulna. Then it goes into a group of short bones, which we collectively call the carpals. Then it goes into more long bones that make up your the metacarpals and the phalanges. And that same bone pattern can be found in all these different mammals. So fossils provide additional evidence of these anatomical unity as organisms descend and modify over time. So Darwin proposed that natural selection could give cause to an ancestral species to give rise to two or more descendants. And this is one of the things he observed with the finches in the Galapagos Islands. How you could have different finches on different islands, but at the end of the day, there was some commonality between them. Now, by no means you have to know this diagram or anything, but if you look, these are different finches that you find in the Galapagos Islands. And one of the things that you notice about finches is it can be different sizes, their beak sizes are different, but they can all be traced back to a common ancestor. So this ancestor split apart over time, leading to a rise of new and new species. As they got to different islands, and those islands, they faced different environmental constraints, and they had to adapt to those new environments or they would die out. The last thing that I want to talk about and go over is something that you should be talking about in lab also. It's just the idea of the basis of science and how scientific studies are approached. And it's the idea that, you go through background information, there's hypotheses, there's experiments, the results, the discussion, everything like that. So let's talk about some of that. And this is going to lead to a homework assignment, which I'll post. So that homework assignment is all about being able to read an article and identify things like the dependent variables, the independent variables. So hopefully lab will help you with that. And I'm not going to talk about this in lecture, but you will have a homework assignment to help you with that. And then of course, if that doesn't help you or you still don't understand it, we'll talk about it and we'll go over it together. But the word science is a Latin term that means to know. So the idea of science, whether it's biological science or chemistry or physics or whatever it might be, is the quest of knowledge. That's what science is. And there's two main types of scientific inquiry. There's discovery science and there's hypothesis-based science. Discovery science is describing natural structures and processes through observation. So this approach is based on going out, looking and observing what's happening in the environment and then analyzing the data. So yes, you still collect data with discovery science and that data can be analyzed. So data are recorded observations or some sort of items of information. And there's two overall categories of data. There's qualitative data, which is descriptions, and there's quantitative data, which is an actual measurement. So quantitative data, you're physically recording or measuring something like how tall something is or how far something moved or the weight of something or the time it took to get from point A to point B. Whereas qualitative data is more of a description. Like, you could say something's tall, something's short. That's a description. And then quantitatively, you could actually measure that and get the actual value for it. But just because it's a number doesn't mean it's quantitative. For instance, if we look at, timing of, let's say, months, like we could say month 1, month 4, month 5, months are more of a description than a quantitative measurement. But if you said hours or days or minutes or seconds, then that's an actually defined measurement. Months are not uniform in numbers, so they're more of a qualitative measurement, just as an example. Now, the term inductive reasoning is looking at how conclusions are drawn through the process of logical induction. And that's basically taking these repeated observations and then making a generalization about it. So for instance, if you were to wake up every morning before sunrise and watch that sun rise, you would realize the sun rises in the east. So we could say the sun always rises in the east, and we're making that conclusion based on observation. So that's called inductive reasoning. However, the thing you have to realize is that's an overall reasonable approach to take. In other words, making this generalization. But in actuality, you don't know if the sun actually always rises in the east. We're only saying that based on the observations we made. But if there's no observation being made, then how do you know it didn't rise from a different location at that specific day or specific time? But overall, we'll make these generalizations based on an overwhelming amount of evidence. So observations can lead to us asking questions and propose hypothetical explanations, and they're called hypotheses. So a hypothesis is a tentative answer to some sort of well-framed question. A hypothesis is an answer that you're trying to predict is going to come out based on your background knowledge. A hypothesis is not a question, so that's number one. The question is not the definition of a hypothesis, it's an answer. So a hypothesis leads to predictions that have to be testable. That's the other thing you have to realize. A hypothesis has to be testable through experimentation. If you can't test it, then it's not a hypothesis. So let's look at an example here. So let's say we have a flashlight and we observe this flashlight is not working. So the question is, why isn't the flashlight working? Well, we could come up with two hypotheses. We could say the flashlight's not working because the batteries are dead. So number one, the batteries being dead is a statement to the answer of that question, why the flashlight isn't working. And that's something that can be testable. You could test to see if the batteries are dead, or you could change the batteries to see if that fixes the problem. But it might not be the only hypothesis. We could also have a different hypothesis. And we could say the flashlight's not working because the bulb is burnt out. Once again, that's an explanation to the question, why isn't the flashlight working? And that's something that's testable. So they're reasonable hypotheses. So the next step would be to test those hypotheses. So we're going to make a prediction that if we replace the battery, the flashlight should work. So what we do is we take the cap off the flashlight, we take out the old batteries, we put the new batteries in, we screw the flashlight back together, but the flashlight still won't turn on. So what we're going to do is we're going to falsify that hypothesis, or we could also say we reject that hypothesis because the dead batteries did not fix the problem. So let's go on and test hypothesis number 2, which was the bulb was burnt out. So now what we're going to do is open the flashlight back up. We're going to take the old bulb out, put the new bulb in. We're going to turn the flashlight back on, and all of a sudden it works. So now what we can say is that hypothesis is supported, or we did not falsify that hypothesis. So you supported hypothesis number 2, which is the bulb burnout, but we rejected hypothesis number 1, which was dead batteries. So here's the question. Did we prove hypothesis number 2? I want you guys to realize you can never prove a hypothesis. You can only support that hypothesis. It can't be proven. Because what you would say here is, well, I proved it. I replaced the bulb and all of a sudden it worked, which means the bulb was burnt out. Not necessarily, just based on our observations that worked, but we didn't prove that the bulb was burnt out. So for instance, when you took the old batteries out and put the new batteries in, maybe you somehow didn't line it up and the bulb was not lined up correctly. Now, when we went to hypothesis number 2 to replace that bulb, we put the new bulb in, it lined up and it actually worked now. But the old bulb wasn't actually burnt out. We didn't prove that. We just said by replacing this bulb, the flashlight now works. So you can't prove a hypothesis. That's a very important concept I want you guys to know. You can support or you can reject it. Now, supporting hypothesis, once again, just means there's evidence that says this is the way it works, but it does not guarantee it always works that way. So as I said, a hypothesis must be testable, and you have to have the ability to falsify it in some way. Hypothesis-based science also uses more than one hypothesis. There can be alternative hypotheses, as you just saw with that example. And as I also just stated, when you falsify a hypothesis or you support a hypothesis, it does not prove that hypothesis. Hypothesis just said, if you support it, that this worked in this scenario, but it doesn't prove that that's the guaranteed only explanation as to what happened. And here's the example I already gave you. So in experimental design, you're going to typically be doing a controlled experiment, and that's where you compare experimental group with a control group. And ideally, there's going to be a variable of interest that you're going to change. And that variable of interest that you're going to change is something I want you guys to identify. So when you look at an experiment, you should be able to tell me what's the control group, what's the experimental group, what's the independent variable, what's the dependent variable. And these are terms you're going to learn in lab. And once again, you'll have a homework assignment for lecture to do that and hopefully help you understand it even better. So in a controlled experiment, you have the control groups that are using to cancel the unwanted effects of the variable. And the controlled experiment does not mean that all unwanted variables are kept constant. Sometimes there's things that are outside of our control. Now, there are limitations to science. So science, you have observations and experiments, and they have to be repeatable. If you can't repeat an experiment to get the same data out of it, then it really doesn't mean anything. Science cannot support or falsify supernatural explanations, which are outside the bounds of science either. So there are limitations as to what science can tell us.